What if I told you that many of the medicines that save our lives are made using the same stuff that powers our cars? Oil. And what if I told you the future of medicine is powered by something much sweeter? Sugar. Today on G Fun Facts Online, we're diving into the quiet revolution bubbling up in our pharmacies, a change that could make medicine not only more effective, but also dramatically greener. For over a century, the pharmaceutical industry has been a pillar of modern medicine. But this success has come at a steep environmental cost. Traditional drug manufacturing is deeply rooted in petrochemicals. This involves complex, multi-step chemical processes that often need harsh solvents, extreme temperatures, and immense pressure. The result is a heavy carbon footprint. Research suggests the pharmaceutical industry's CO2 emissions are staggering, with some estimates indicating they are greater than the automotive industry's when measured per $1 million of revenue. We're talking about an estimated 52 megatons of CO2 pumped into the atmosphere annually. And that's before you even consider the hazardous waste generated, the massive water consumption, and the non-degradable pollutants that can contaminate our ecosystems. It's the bitter pill behind the life-saving cure. But a change is brewing, quite literally. Scientists are rewriting the playbook for drug manufacturing by turning to one of nature's most fundamental building blocks, sugar. This paradigm shift, often called sweet medicine, harnesses the ancient power of microbial fermentation. By programming tiny cellular factories, microorganisms like bacteria and yeast, to perform complex chemical syntheses, the industry is creating a more sustainable and efficient future. This isn't a distant dream. It's a rapidly advancing reality. So how exactly do you brew a life-saving drug from sugar? The process starts with two microscopic workhorses, the bacterium Escherichia coli, better known as E. coli, and the baker's yeast Saccharomyces cerevisiae. You might know them from biology class or from baking bread, but in the world of biotech, they are superstars. Their rapid growth rates and well-understood genetics make them perfect candidates for industrial-scale production. In their natural state, however, these microbes are programmed for survival, not for mass-producing a specific drug. Only a tiny fraction of the sugar they consume would naturally be converted into the molecule we want. That's where the magic of modern science comes in, through two key disciplines: metabolic engineering and synthetic biology. Think of metabolic engineering as rewiring the cell's internal assembly line. Scientists act like city planners for the cell's metabolism. They can overexpress certain genes to boost the production of key enzymes, effectively creating more workers on the important part of the assembly line. They can also delete genes that lead to unwanted byproducts, closing off inefficient side streets and redirecting all the traffic toward the desired product. Synthetic biology takes this a step further. Instead of just tweaking existing pathways, it involves designing and constructing entirely new biological parts and systems from scratch. Using powerful gene editing tools like CRISPR-Cas9, scientists can now insert, delete, or modify genes with pinpoint accuracy. It's like having a word processor for DNA. They can design and build custom microorganisms tailored for one specific job, producing a single, pure pharmaceutical. Once the microbe has been engineered, the manufacturing process begins. This is called fermentation. The engineered microbes are placed in large, sterile stainless steel tanks called bioreactors, which can be larger than a city bus. Inside, they are fed a carefully controlled liquid diet, primarily composed of a sugar source like glucose or sucrose, along with other essential nutrients. Parameters like temperature, pH, and oxygen levels are meticulously monitored to create the perfect environment for the microbes to thrive and produce the drug. After the fermentation is complete, the next phase, called downstream processing, begins. This is often the most challenging and expensive part of the whole operation. The target drug must be separated and purified from the complex soup of microbial cells, leftover media, and other byproducts. This involves multiple stages, like harvesting the cells, breaking them open if the drug is made inside, and using a series of purification techniques like chromatography to isolate the final pure drug molecule. So, what kinds of medicines can we find on this sweet menu? The list is longer and more impressive than you might think. The story of industrial fermentation in pharma actually began with antibiotics. The discovery that the penicillium fungus could produce bacteria-killing penicillin was a medical revolution. The challenge was making enough of it. During World War II, engineers adapted deep tank fermentation methods to mass-produce penicillin, feeding the fungus a diet rich in sugars. This technology is still the foundation for many antibiotics produced today. Fermentation is also a critical technology in producing modern vaccines. Many vaccines, including those for hepatitis B and human papillomavirus or HPV, are made using engineered yeast cells. Perhaps the most significant application of sugar-based fermentation is in the production of biologics. These are large, complex protein-based drugs that are difficult, if not impossible, to create through traditional chemical synthesis. The most famous example is insulin. Before the biotech revolution, insulin for diabetics was extracted from the pancreases of pigs and cows, which could cause supply issues and allergic reactions. Today, the vast majority of the world's insulin is produced by fermenting genetically engineered E. coli or yeast. This technology also underpins the production of monoclonal antibodies, a class of drugs that has revolutionized the treatment of cancer and autoimmune disorders. While these are often made in mammalian cells, microbial systems are becoming more and more common. The technology is even allowing us to make complex small molecule drugs that were previously only available from unsustainable natural sources. Take artemisinin, a potent anti-malarial drug. It was originally extracted from the sweet wormwood plant, but the supply was often unstable and subject to weather and politics. In a groundbreaking achievement of synthetic biology, scientists engineered yeast to produce a precursor to artemisinin from a simple sugar feedstock. The pharmaceutical company Sanofi now uses this microbial fermentation process to manufacture a significant portion of the world's supply. In a similar feat, researchers have successfully engineered yeast to produce opioids like hydrocodone directly from sugar, a process that could one day offer a more secure supply chain for these critical pain medications, which are currently derived entirely from the opium poppy. The benefits of this sweet revolution are profound. The most celebrated advantage is the reduced environmental impact. By swapping finite fossil fuels for renewable feedstocks like sugarcane, corn, and in the future even non-edible agricultural waste, the industry can slash its carbon footprint. Bio-based production processes generally occur under much milder conditions, in water, at or near room temperature and normal pressure. This is a stark contrast to the harsh, energy-intensive conditions of petrochemical synthesis, saving energy and reducing the need for hazardous solvents. The economic incentives are also compelling. For many complex molecules, microbial fermentation is a more cost-effective manufacturing method. Using cheap, renewable feedstocks can significantly reduce raw material costs. It also creates more resilient and stable supply chains, insulated from the geopolitical instability and climate events that can affect plant-derived or petrochemical-based resources. But perhaps the most exciting advantage is the potential for innovation. Synthetic biology platforms allow scientists to rapidly design, build, and test new production pathways for both existing drugs and entirely new molecules, dramatically shortening drug development timelines. However, the path to a fully bio-based future is not without its sour notes. There are significant hurdles. One of the biggest is scaling up. A process that works perfectly in a small 10-liter lab fermenter may behave completely differently in a 100,000-liter industrial bioreactor. Maintaining consistency and managing things like oxygen supply and heat removal becomes exponentially more difficult. Then there's the regulatory maze. Using genetically engineered microorganisms to produce drugs introduces new layers of complexity. Companies must prove to regulators like the FDA that their final product is completely safe and pure, a process that is both time-consuming and expensive. finally, the economic equation doesn't always favor the bio-based route. The petrochemical industry has had a century-long head start, with highly optimized processes and massive economies of scale. For some simpler, high-volume chemicals, it remains the cheaper manufacturing option. Despite these challenges, the future is brewing, and it's being accelerated by powerful new technologies. Artificial intelligence and machine learning are transforming every stage of the process. AI algorithms can analyze vast data sets to predict how genetic modifications will affect a microbe's performance, allowing scientists to design better cell factories faster than ever. In one incredible example, I was used to design the mRNA sequence for Moderna's COVID-19 vaccine in just 2 days. This AI-driven design is powered by robotics and automation, which can build and test thousands of engineered microbial strains in parallel, generating massive amounts of data to feed back into the AI. We're also looking beyond simple sugars to the next generation of feedstocks. Scientists are developing enzymes that can efficiently break down tough, non-edible plant matter like wood chips and cornstalks into fermentable sugars. An even more radical vision involves engineering microbes to consume C1 compounds, things like methane, or even carbon dioxide captured directly from the atmosphere. Imagine a factory that literally turns industrial pollution into life-saving medicine. This is the vision of a sustainable, circular bioeconomy. A future where the medicines that heal us also help to heal our planet. The sweet medicine of tomorrow is no longer a distant dream. It's being brewed in labs and factories around the world today.